Hi, and welcome to second lesson of 17.2. We're going to look at a few different kind of crosses today, and actually the probably three last types of crosses that we will be doing will be in this lesson. So you want to pay close attention to how we do these, and feel free to pause the video and try some of these out as we go through them. Even if you see I'm about to work it out, you could stop and try it for yourself. Thomas Morgan is profiled here because he was very famous for his studies with the fruit fly and the crosses that he did to find out about another kind of genetic mode of inheritance. So we talked about how there's, the principle of dominance, and there's the dominant recessive alleles, the regular crosses with that. We talked about codominance and intermediate inheritance. But when Thomas Morgan was studying flies and he used flies for all of these reasons here. They reproduced very quickly and had easy to see phenotypes. And what he realized was there was the ones he was crossing, the white eyes and red eyes. He crossed the red and the white, and he got 100% reds, crossed his reds, and he figured he should have a 3-to-1 ratio, familiar cross. He should have a 3-to-1 of red to white. But what he didn't understand is that what he actually realized the results of that second cross was that he said that there was half of the males with white eyes and half of the males with red eyes, and all the females were red. And so this seemed coincidental maybe the first couple times, but every time he did this cross, the same thing happened. So what he realized was that the genes he was studying were genes that were on the sex chromosomes, and females had 2 of those, XX, and males had 1 X because they also had a Y. So if it was a gene on the X, then males would only have the 1 copy, whereas if it was females, they would have 2 copies. So this is what we call X-linked traits or sex-linked traits, traits that are on the sex chromosome. So if you look at what males have, they have 1, 2, 3 homologous pairs, and the 4th one is the sex chromosomes. That's what the males would look like. And the females, there's their 1, 2, 3 homologous pairs and 2 X's that are the same size. So just as a review, your diploid number for these Drosophila would be 8 chromosomes. And there you go. Okay, so we mostly talk about X-linked traits, the ones on the X chromosome, in pretty much all of the crosses that we're going to do. The ones on the Y would obviously only be on males, and they would be on the Y chromosome that's given to their sons. So it's a little bit too easy, and there is only a few traits that we talk about that are on the Y chromosome, and basically it's the testes determining factor for male hormones, and actually a gene also for hairy ears that's found on the Y chromosome. On the X, though, there are quite a few, which I'll show you in a little bit in a future slide. But when we write down our genes for sex-linked crosses, we will write down this. This didn't work out too good. So we write down the gene that's on the X, and we don't write anything on the Y because we're looking just at the X. And this is what the female would look like. It has 2 X's, so you put the gene that she has for each X on there. So if they say it's a homozygous red-eyed female, it would look like this genotype. If it was a white-eyed male, it would look like this genotype. When you do that cross, you do this the same way that you do other crosses, you just put them in a wee little Punnett square. And this first one would be a female with red eyes. Next one, female with red eyes. Next one, male with red. And next one, another male with red. And you notice I never put anything on the Ys. The Y is not carrying any gene that we're studying when we do the sex-linked traits. We're looking at just what genes are carried by the Y. But basically, this is showing you that all their offspring would have red eyes, but the females would be heterozygous and the males would just be regular red-eyed. Okay. Incidentally, when we figure out our phenotypes, we do separate males and females. So this would be a 1-to-1 ratio of red males and red females. So males and females for the sex-linked crosses are different phenotypes. Okay. So to do the F2, what they did is they took one of the red-eyed males, crossed it with one of the red-eyed females that's heterozygous, and you guys can try this cross on your own just to make sure you are used to working with your sex-linked genes. So there's two girls who are red eyes but different genotypes, and here's a red-eyed male offspring, and here's a white-eyed male offspring. So incidentally, all four of these are all four different genotypes. These are all different genotypes, but physically all girls have red eyes, and half the boys are red, half the boys are white eyes. So in that case, for phenotype, we have a 1-to-1 ratio. For genotypes, there's four different ones, so it's a 1-to-1 to 1-to-1 ratio for genotypes. Okay, these are pretty easy. One human example is color blindness, in which some cones have issues, and in humans it is normal to have the regular vision, and then I used C for color blindness in this case. That's what they were using. So for the recessive, the colorblind gene in this case, I used the lowercase c, but don't forget to put that little line on it here so that you don't mix that up. And so when they say the colorblind heterozygous female, that means she's carrying that colorblind gene. And for the male, he is colorblind, so he's got the gene. So set up your cross. You guys can pause the video and give this a try. But basically, it's no different than what you've been doing before. And you can see where putting the little line over the C is helping to keep you organized as to what your offspring are here. And what you notice is that the girls are, half of them can see regular and half of them are colorblind. And for the boys, half of them are regular vision and half of them are colorblind. So again, we have 4 different genotypes, and in this case, 4 different phenotypes as well. Hemophilia, another gene in humans that's on the X chromosome, sex chromosome. And so hemophilia, normal is I have the big H, and hemophilia, the condition, is the recessive little h in my legend here. So the hemophiliac male is one that has the little h here on the X chromosome, and remember, don't put anything on the Y. And this is a homozygous dominant lady. And so what we notice here, oops, is that, pardon me, just make that a little neater, is that the first girl is heterozygous, so she won't be hemophiliac. This second option is, or second offspring, is normal male. Here, their other daughter would also be a carrier, still not hemophiliac. And so there you go. You've got all their kids would not have hemophilia, but their girls would be carriers. One thing that's interesting over here is, no, that's not what I was going to say, is that sometimes when you have two parents that don't have a sex-linked condition, if it is a male that does not have a sex-linked condition, for example, crossed with a female that doesn't exhibit it, but she's carrying it, interestingly, it will be her that will give it to her sons. So you can see here, this girl fine, this son fine, this girl a carrier, and here we go with a son who has hemophilia. So two parents who maybe don't have a sex-linked condition, if they do show up with it, it will always be the mom who gave it to the sons because she was hiding that recessive and gave it to her sons. And this also is why in sex-linked conditions like color blindness or hemophilia, it is more likely to show up in boys. So conditions that are very prominent in males are often conditions that are on the X chromosome, because those males just have the one chromosome. So if they got that recessive, they exhibit that recessive trait. And this is a picture of the X chromosome and all of the disorders that could be on there. And there's a very long list of different disorders that they've mapped out to be on the X chromosome, which is very interesting. And hemophilia, there's two types are right there. Color blindness, the ones we've talked about, and many others. All right, so the Y chromosome here is, this is actual picture of a replicated, there's a little Y, this is X, it's just much bigger, it's replicated in this picture. But they mention here that the Y has hairy ears as well as testes determining factor that activates around 7 weeks and starts producing testosterone, therefore male sex characteristics, at around 7 weeks. So prior to that in fetal development, you can't really tell if it's a male or female little embryo. All right, so we are going to talk about, I think you guys can confidently do your sex-linked crosses that are in your practice. And we have this little interesting thing about bar bodies, and this is just for the genes on the X chromosome. For some reason, sometimes only one of those, and this is just for the number 23 pair, for the sex chromosomes, sometimes only one is active and the other one balls up and it becomes inactive, and it's called a bar body. This is not a major outcome in your curriculum, but it's kind of interesting to see how what can happen because of that dormant X. And it says it's kind of random in the body which X is dormant. So if they have something lethal, this is pretty cool. If there's a lethal X-linked gene, but it's only active in half the body cells, then that female can actually escape dying from that condition. So the other one is this anhydrotic ectodermal dysplasia, where there is excessive sweating, sweat glands, and if for the dominant, but the recessive is no sweat glands at all. And so interestingly, because it's on the X chromosome, women with this, it has to be women because they have to have two X's, they may actually have, so parts, let's say some is normal, some is recessive, that is no sweat glands. They can have this appear as patches of areas that on their body that have sweat glands active and patches that don't. So that's an interesting thing about the inactive or dormant X. It's random which areas of the body has this one active, and random which has this active. So this brings us to calico cats, and they find that the coloration is actually in an X chromosome, and so the only cats that would be like the male ones, they would have either the black gene or the orange gene. So the male cat is either black or orange. But if there is a mix, the calico cat, it's because it's a female who has that color on, those colors on their X chromosome, and because patches are dormant, there are patches that end up being orange because the black is dormant, and patches that turn black because the black is dormant. Now the white would be fur color that's found on a different set of chromosomes, but for the black and orange part, it's very likely that the calico cat is a female. And so in this cross, we're looking at the orange mom and the black dad, and how they can end up having the calico girl, or they could have orange boy or orange boy. So it's interesting that the mom is orange and her boys are going to be orange, and dad is the black cat, and they're going to have all calico girls. So kind of cool to try one of those out. And multiple alleles. We are not going to talk about or do crosses that have 22 genes in them like clover, but multiple alleles is when there's a gene that has multiple versions of it, not just two like tall, short, or green, yellow, or round and wrinkled. It's where there are many different alleles possible for that gene. And one example is this rabbit question, where there's 4 for this certain species of rabbit. There's 4 different colors that they can be. They can be agouti, they can be chinchilla, and they can be Himalayan, or they can be albino. Now, I wrote this out like this because what we do with multiple alleles is we check out the hierarchy of dominance that exists. And hierarchy of dominance means that the one at the top here, agouti, is stronger than any of these other colors. So if it shows up with any of these alleles with it, it will still be agouti colored. Whereas chinchilla here, it can be chinchilla with 2 of these. It can be chinchilla if it's found with either of those because it's stronger than those. And same thing with this Himalayan. It's Himalayan if there's 2 Himalayan genes, or it's Himalayan if it, I think I was supposed to actually write that C to the H. Himalayan if it's carrying the albino gene, and the only way for it to be albino is for it to have 2 of these. So you're just basically looking at the hierarchy of dominance to see what those babies would look like. In terms of this rabbit, you could say I've got a rabbit with this dominant agouti color carrying maybe a chinchilla gene. And what happens if I have a chinchilla gene and it's carrying or hiding a albino gene? And so when you do a cross like this, you just want to put the more dominant one first. So if I'm going to do this one with the dominant agouti and this chinchilla, I want to put the agouti first. That's a agouti colored baby. This one has agouti and the albino, but it will turn out to be agouti. So these 2 will look agouti, even though they're carrying a gene or allele that's more recessive with it. This one is 2 chinchillas, so obviously be chinchilla. And this one, I'm going to write my chinchilla before I write my albino, because albino is more recessive. So these 2 individuals, we've got a dominant agouti color and chinchilla rabbit, and they actually end up with 2 that are agouti like one parent, 1 that's chinchilla like one parent. Sorry, these 2 are chinchilla. So yeah, there you go. 1-to-1 ratio. That's how we do it. Okay. Blood types might be something you remember from Bio 20, that type A people can have 2 A's on their 2 chromosomes. They're type A. They can still be type A even if they're carrying an O. Type Bs can be homozygous B, or they can be heterozygous B because they're carrying an O. They're still both, that would still be both B bloods. There's type AB blood and there's type O blood. So these are basically codominant, A and B. So if they show up together, the person is type AB. Basically, type A's have type A antibodies on their red blood cells. Type B's have a different one, a B antigen on their red blood cells. AB has both type antigens on their red blood cell, and O has none. So AB is considered codominant because both of those will show up, whereas A is dominant over O, because if you have A and O, it's going to be type A. If you have B and O, it's type B. And so it's an interesting condition where A and B are codominant and type O's are recessive to A's and B's. So what happens with, in your diploma questions, you'll notice they, don't ask me why they choose a letter I, but anyways, they choose a letter I, as you see here, to put the gene types on. So for they're just showing you that you could have type A that has, is homozygous with A's, or you can have A's that are carrying a recessive O. And if they're carrying a recessive O, the way that they show this is that they still have the A, but they put the O in a lowercase I just to show that the O is recessive to the A. And they'll do the same thing for the B. Now, what I'm going to say is that I particularly don't like to write all the I's, and you'll notice with other crosses that as long as you have a legend that you follow and make your own symbols, you'll be fine. So when we talk about someone that's homozygous A, I will be writing this. If they're AB, I'll be writing this. If they're carrying an O, I'll do this. If they are OO, then I'll be writing that. But you may see a diploma question that uses those letter I's and just puts the BI means B, carrying an O, and II means that it's OO. So that I system is just, to me, another thing to write, so I never write those down. But you guys can figure that out, what it means. Okay, so this means B and B. This means B and O. This means A and A. This means A and O. This means O and O. This means A and B. Okay, so when we look at type A blood, it's got the one antigen for A's, B has antigens for B's, AB has both, and O has none. There is another factor, the Rh factor, that is, when it's there, it's dominant. So Rh positive people have this extra antigen on their blood cells, and they wrote it as a box here. So this one here would be AB positive because they have that extra antigen marker. This O has it here, so this would be O positive. If they don't have it, they would be O negative. And majority of people are, I think 85% are dominant and have it, and maybe the rest, 15%, are Rh negative and don't have that extra antigen. This is important when you're donating blood, I guess, because if you have no Rh factor on your blood cells, then you cannot accept blood that does have it, because your immune system would definitely fight against that, because they'd notice a foreign antigen marker. So we definitely, keep that in mind as a incompatibility potential. All right, so when we do these to practice doing blood types, these are really, really simple. But a homozygous person who's a B, it just means that both of their alleles are Bs. And if they cross them with a type AB, you just simply do that. And you can see what their kids are going to be. 50-50 ABs and Bs. Number 2, with paternity questions, they're saying mom is a type B. So well, you don't know what her second gene is, and dad is a type O, but you can check out whether it's possible for them to have an AB baby, and it is not, because if she is B type, she either has a B here or an O here, and that means that this A could have not come from this dad. So you can definitely answer the question that he is not the father. For this next question, if mom is BB and the baby is AB, which dad could give the genes for that baby? And it would have to be one that gives this A. So definitely Don is the daddy. So Don with his AB could be the dad of a AB child. So that's how you check those out. I gave you some to practice in your notes. You can check those out. This is one of those hierarchy of dominance. There's 4 different colors of eyes in fruit flies. This didn't come out so nice on my PowerPoint here, but E1 is the one that's the red one, and it is stronger than eye color number 2, which is apricot, which is stronger than eye color, I think I just wrote my arrows the wrong way, pardon me. Stronger, yeah. E3, which is the honey, and that is stronger than the white color eyes, number 4. So with this hierarchy, what you can do is be able to try things like, here's a red-eyed fly that's carrying a white eye allele, and if you cross that with honey color with carrying a white eye allele, this is really a red-eyed fly, because you don't even in the color, it's going to be the color of the most dominant on your hierarchy, and this fly is going to be a honey-eyed color fly. But if you cross these, you can see that when you have to join the E1 and 3, you're going to have to choose, first of all, to put the one that's higher up on the hierarchy. So the one before the 3. This is a red-eyed fly. Over here, the one before the 4. That's also a red-eyed fly. Over here, the 3 before the 4. That's a honey eye. And over here, the 4. And so what happens here is half your babies are red eyes, and a quarter of them are honey. And then you end up with a quarter of your babies a color that neither parents are, but that both parents are carrying. So you get a quarter white-eyed flies. And these are easy to do. You just have to keep glancing over to the hierarchy and making sure that you write them properly. So this got all messed up, but you guys can try an E2, E4 crossed with an E1, E3, and see that you get these offspring and do the cross for yourself. Okay, just to wrap up here, something continuous would be a trait that's something like height. So they're on a continuum, or weight, many different eye colors, many different skin colors. We're not going to do crosses with those, but they're called continuous. And polygenic, there's also many traits controlled by many genes, not just one, so they get complicated, and we're not doing any crosses with that either. They're just showing you here that if you have more dominant genes, they will be like here's 2 genes involved, but the longest ears of corn are ones that have more dominance, and the shortest ears of corn have more recessive alleles. And same with pigmentation, the more dominant alleles for the skin color, the darker the skin is, and all really pale genes, really pale, and most of the population is a mix of half and half, dark alleles and light alleles, but that's an extra stuff. Okay, last concept for today, for this lesson, is epistasis, where some gene gets masked, and this would mean just reading the story and seeing what their condition is for the epistasis here, and writing a legend that will help you know how to do the question. And for this, your question just says that B is that black color, and little b is the brown color, but if there's a capital or dominant W, there will be no color at all. And so there'll be white coats on these dogs. So when you look at the genotype, because there is a big W here, it's a white dog no matter what their B's are. And in this case, because it's lowercase w's, the recessive, this comes out as a black dog. So the coloration only comes out if there's recessive w's there. So trying the cross that they're describing here, I just made my 4 alleles for this. It's a regular dihybrid. This one will end up having 2 alleles. And do the cross. You guys can try this for yourself. But basically, because these all have big W's, they're all going to be white dogs. And 4 out of the 8 will be white dogs. And these all have homozygous recessive W's. So you get also 2 black dogs and 2 brown dogs that show up, even though there's no brown parents. They both were carrying brown, so we get brown dogs. And so this is like ending up to be, this was 4 of something, 2 and 2, so it's going to be a 2-1-1 ratio when you reduce it. So it comes up with some interesting ratios. Okay, the most question you're going to see in your notes, but basically there is, in this case, the recessive 2 little c's that mask the color of the mouse, which is why these are all white mice. And if there's a dominant C, they will be either black or brown mice. So it's just a different rule for different epistasis examples. You just have to read the rule, make yourself a legend, and they'll be easy to do. Okay, you guys can read some interesting environmental conditions that give these, for example, cats darker coloration where they're colder. So environment has something that will add to their phenotype, and where they're warmer, they turn more white. It's just some interesting examples. You guys can check those out, and I will say bye for now. Let's go practice these cross.